Welcome back, everyone, or welcome along to anyone who's just joining us for the first time today. I'm really happy to introduce our next session, our science deep dive in this conference. For anyone who is at all familiar with the Rivers Trust, you'll know that science and evidence is really crucial to all of the work we do. So I think that's why it's really important that we have a focus on it here in our conference today, but also that I can now introduce the chair of this session, who is our technical director, Michelle Walker. And yeah, Michelle, you're the perfect person to be introducing this session. Topics very close to your heart. So yeah, take it away.
Thank you very much, Becca. Yes, hello everybody. As Becca said, being evidence-led is a core value of the Rivers Trust and the Rivers Trust movement. We've got a growing technical team in the Rivers Trust, but also, in our member trusts who really use evidence to understand the issues and the causes of those issues fully, and to target the right interventions in the right place, and to monitor the effectiveness of what we're doing, whether things are working, and take an adaptive management approach, but also to be able to communicate and advocate for the right changes as well. We've got to be evidence-led in that. So we partner with the scientific community to ensure that we can apply the latest research and make an impact for our rivers. So in this session today, we're going to have two presentations from the scientific community and learn about some of the underpinning science behind the causes and impacts of blue-green algae, and the emerging science in how we can monitor and test for these issues, and also the role of communities in helping to do that as well. That's a subject that's very close to my heart, and how we can all help track and report these problems, but also some of the innovative mitigation measures that are being developed, including around Lough Neagh that we've heard about today. So I'm delighted to introduce firstly to you Katrina Campbell, who is a professor in food security and diagnostics at Queen's University of Belfast. So Katrina is a fellow of both the Royal Society of Chemistry and the Royal Society of Biology, and a member of the Irish Toxicology Society and the International Society for the Study of Harmful Algae. Her research really focuses on identifying the known and emerging threats within the food supply chain. So really going from environment to farm to fork to waste. And she designs state-of-the-art rapid methods of detecting toxins, chemical contaminants and harmful organisms, and really all about focusing that on enhancing water quality and food safety and sustainability to benefit both wildlife and human health, with an outlook on increasing food security. So I'd like to invite Katrina to take us through her presentation, and to remind people, please put any questions in the Q&A and we will have the chance to address those in a Q&A session at the end. Thanks, Katrina.
Thanks, Michelle, for that very nice introduction. I'm just going to go on to slideshow now. Maybe if I can get a thumbs up that the slideshow is good.
Yeah.
So what I would like to talk about today is bloomin' blue-green algae, about their detection and mitigation. It's a little bit about what they are, where they come from, and how we can detect and possibly get rid of them. So cyanobacteria, as you know, are a group of photosynthetic bacteria widely distributed in various aquatic habitats, oceans, seas, rivers, and lakes, but also terrestrial habitats, for example, moist soils. They may live singly or in colonies by forming filaments or spheres with other cyanobacteria. They are very much an important oxygen producer of the planet, so they're a necessity for the planet. They have existed for billions of years in the early extreme environments before plants and animals even emerged. And early cyanobacteria played a role in the great oxygenation event by releasing oxygen as a byproduct of photosynthesis. And then there is the endosymbiotic theory, whereby they form symbiotic associations with other organisms. And then the endosymbiotic cyanobacteria turned into plastids, like chloroplasts, of the photosynthetic eukaryotic cells. So these cyanobacteria have been around for a long time, and they're very much part of our natural environment. In terms of the major taxonomic orders of cyanobacteria, there are 5 main orders. They have different general features, and there's some examples of these particular orders. For example, Crocales, you've got Microcystis and Anaphanocapsa, and they have different shapes within the different features, which actually helps to identify them. And most of the identification would be done then by light microscopy. When we have cyanobacterial blooms, this tends to occur when the conditions are favorable and allowing the cyanobacteria to grow exponentially. And the phenomenon is called an algal bloom. And you can see the picture here taken of Loch Ness, this kind of large algal bloom just encroaching towards the land of the lake. Now, algal blooms occur especially in aquatic environments that are warm, stagnant, or slowly flowing, as these areas tend to accumulate high concentrations of blue-green algal nutrients like phosphorus and nitrogen. You've also got marine and freshwater cyanobacterial blooms, and these are scum-like and blue-green in colour, as you can see in the picture. Water samples of algal bloom sites indicate the most common cyanobacterial genera. They tend to be Microcystis, Anabina, and Planktothrix. The cyanobacteria can also produce toxins, and particularly those that occur in fresh water. They are distinct cyanotoxin and non-cyanotoxin producing organisms. The organisms involved in toxin production include an estimated 40 genera. And cyanobacteria can produce a wide variety of cyanotoxins, which tend to fall into 3 broad groups: cyclic peptides, alkaloids, and lipopolysaccharides. Now, most cyanobacterial toxins are intracellular. They stay within the cell, and the toxins then tend to be released as cells die or break open upon treatment with chemicals, for example, the use of copper sulfate. The toxin content is highest in cells at temperatures between 18 to 25 degrees centigrade, and the toxin content becomes lower at temperatures less than 10 degrees centigrade or at very high temperatures above 30 degrees centigrade. The cells have been found to have high toxin levels when grown at high and low pH conditions, and toxins content can be higher in nutrient-rich waters with increased phosphorus or nitrogen. So really, the toxins can really be found to be higher when the waters are richer in nutrients. In terms of the health effects and possible exposure pathways, these particular toxins can act as neurotoxins, liver toxins, cell toxins, and also skin irritants. The toxins may result in animal deaths with large losses among wildlife and domestic animals. They can be involved in human poisoning cases, which have occurred globally. Children tend to be more susceptible to toxins due to their smaller body size and the potential for more incidental ingestion and response to symptoms. You can have chronic ingestion through contaminated drinking water, the ingestion of water, inhalation of droplets, or contact with nasal mucous membranes through recreational contact. And you can also have dermal contact with toxins through bathing or recreational activities, such as wading, swimming, skiing, or canoeing. There's also some concern about the consumption of toxins within fish and also shellfish from contaminated waters. And water used as recreational bathing waters and human consumption should be regularly monitored for cyanotoxins if cyanobacterial blooms tend to occur. Now, this particular table shows an overview of the freshwater toxins, their producer, the toxin, and the health effects. So in relation to the toxin, these can be classified due to their chemical structure. And some of the main toxins that can be found are microcystins. Then we can have some of the alkaloids, which would be saxitoxins and anatoxins and cylindrospermopsins. And then we can have other toxins such as polyketides, ethylatoxins, and amino acids as well that can be found. And these would be non-proteinaceous amino acids. We can also see the genera of the species that would be producing these particular toxins if found. And we can also see where the vector would be in terms of any human or animal intoxication, perhaps drinking water, irrigation water, or recreational waters. We know in a lot of cases the short-term health effects for a number of these toxins. In terms of, you might have gastrointestinal effects, some liver inflammation, sometimes they can actually lead to death if they're present at very high levels. And sometimes that's where we would see then perhaps animal deaths close to lakes or ponds that are contaminated with cyanobacteria. And these would be from toxins like anatoxin or saxitoxin, that would be very potent. In relation to the long-term health effects or the chronic exposure of these particular toxins, there's a lot of uncertainty and a lot we don't know. We think there are links because they're tumor promoters in terms of certain cancers, particularly liver cancer or liver failure. We also think that some of these toxins can lead to cardiac arrhythmia. Some of them also can lead to different tumors, such as skin tumors, and they can also be known carcinogens. In some cases, the toxins have actually been linked to neurodegenerative diseases, such as Alzheimer's and Parkinson's disease. If we look at some of the cyanobacteria in a little bit more detail, these would be some of the most commonly found globally, and also within our water body in Loch Ness. We've got Microcystis aeruginosa, and globally they produce microcystins, and these are the most common detected cyanobacterial toxin in fresh and brackish waters. And microcystis is one of the biggest problems that we have in Loch Ness, also with the microcystin toxins. These particular toxins are water soluble, and most microcystins cannot penetrate directly through plant or animal cell membranes, so their uptake is into cells occurs through transmembrane organic ion transporting polypeptides, or through proteins that really take them through the cell. The liver is the ultimate target organ for toxic effects, because the liver is usually the main organ for detoxifying different substances that would be ingested in the body to process it and eliminate it from the body. Now, microcystins are highly toxic with low doses required for lethal effects, and there is some evidence that microcystins can act as tumor promoters as well. And some studies have actually found in fish fillet globally the range of around 300 parts per billion of microcystin. And when we consider the action level or the considered action level as part of the Water Framework Directive as 1 microgram per litre, you can see that having around 300 parts per billion is actually a very high level. Another species of cyanobacteria would be Cylindrospermopsis raciborski. And as more water bodies are tested and analysed for the presence of Cylindrospermopsin, these toxins are found on a global scale. There are certain strains of Cylindrospermopsis anabina, anaphysomenon, and others have been determined capable of producing this particular toxin. The main target of toxicity is the liver, and other organs such as the thymus, kidneys, lungs, intestinal tract, and heart can be affected. Now, Cylindrospermopsin was reported to be a possible tumour initiator, and symptoms of Cylindrospermopsin toxicity in cattle have been reported as weakness, anorexia, pallor of mucous membranes, and even diarrhea. Now, several incidents of death in livestock have been attributed to this particular toxin on a global scale. Anabina flossaqua produces anatoxins, and as you can see, anatoxins are neurotoxic agents produced by a variety of cyanobacterial organisms. There are alkaloids, which are capable of transformations into toxic byproducts. And anatoxins are stable in dark waters, but are susceptible to photochemical degradation, and breakdown is further accelerated by alkaline or high pH conditions. Now, anatoxins have been implicated in numerous animal and wildfowl poisonings, and symptoms include death, which can occur in minutes, and particularly in dogs that might be bathing in contaminated waters. There's no information can be found on chronic exposure to anatoxins, and really their exposure, because they're such a potent toxin, can, if you're exposed, you will know fairly quickly that you've been exposed due to the symptoms that they would elicit. Now, if we look at Lough Neagh, we can see in Lough Neagh the number of blue-green algae locations that have been detected right out to the marine environment. And this was done in 2023. And you can see the typical blue-green algal scum that occurs close to the shores and also then further into the water body. Now, because Lough Neagh is such a large loch, that's part of the difficulty in any of the mitigation methods that may be there. Now, as we can see here in the pictures, it's very easy to see without monitoring to any great extent or using highly fancy techniques that there is a cyanobacterial bloom present. But what we should be doing is monitoring all year round to try and get a better understanding of where the cyanobacteria is coming from in terms of where it occurs first in a particular period and how then it depurates and disappears from the environment in the colder winter regions, to at least a certain extent, if not completely. Some colleagues of mine published a paper in Environmental International, and part of the paper they had this lovely schematic. With the lovely schematic, you can see all of the things or all of the elements that impact Lough Neagh in Northern Ireland, right through from agriculture in terms of lowland cattle grazing, sheep grazing, slurry, fertilizers, commercial eel fishery, and also then forestry or urbanization in terms of a town located very close to the lake, and also then where the water treatment works and combined sewer overflows are as well. There's also dredging that goes on within Lough Neagh, which again may have an impact in helping, the overall environment and the overall ecosystem in terms of cyanobacterial production. We can see in terms of the surface waters, Microcystis, actinobacteria, candidus, and another candidus species was detected. And we can also see then in terms of any of the algal mats that we had different bacterial species such as Clostridium, E. coli, Pseudomonas, Yersinia, Cloacina, Salmonella, and Clostridium. And particularly around where the sewer overflows would be. And the main toxins that would be present would be Microcystin RR and Microcystin LR, and some of the anabenopectins, which actually give that characteristic odor or unpleasant odor from algal blooms. You can see the wind direction from the west to the east, and you can also see the July rainfall and quite heavy July rainfall that actually happened usually after we have a beautiful spring like we're having today, where the sun has been at its highest. We've got lots of plant production, plant growth. Then with the July rainfall, we would have quite a lot of rain running nutrients and effluent perhaps into the loch and creating then the algal blooms. We also have Lough Neagh flies. We have wildfowl that can be affected. We can have animals that can be affected that might be swimming or grazing near to the lake. And we might also have potential contamination of toxins within the eel industry. So it is a loch that we have to really protect. And we also have endangered pollen that we need to consider as well. There's legacy phosphorus within the sediments that we have to consider as part of our overall ecosystem. And of course, then the recreational activities in terms of water sports, fishing, that may also go on in relation to the whole overview of the ecosystem and a holistic approach. So when we consider any water body, we have to consider all the inputs and possible environmental factors that may help to cause cyanobacterial blooms, or that could have in future a role of mitigation of some of those blooms. And when we look at one of the big factors is, whether we like it or don't like it, is our climate change. And if you look at the bottom graph here, you can see the climate change that has been documented since 1940 to 2022, as all the grey squiggles going across in the year. And from 1940 to 2022, we've gradually got warmer. When we look at 2023, we can see there's a big increase in temperature and a big spike in temperature, usually in around June, July time, and right through then to September, October time, even right through to December time, that was warmer than any year previous. We can also see the same trend was beginning in 2024, where these have been the two highest temperature trends documented in history. So with this new climate, we're getting more hot weather, more hot spring weather, but we're also getting increased rainfall. And cyanobacterial blooms are intensified by some of this anthropogenic eutrophication of aquatic habitats. Now, whether this be rising temperatures, vertical stratification, increased carbon dioxide in the atmosphere, or high concentrations of phosphorus in eutrophic lakes and oceans, these can cause a cyanobacterial population to grow exponentially. If you've got the addition of other nitrogen nutrients, phosphorus nutrients coming off the land, it will also help that cyanobacterial growth. In terms of detection, recreational bathing waters come under the World Health Organization, and light microscopy is used for the identification of cells. Now, it's used for the identification of cells to identify what the bloom actually is. In terms of risk level, there's relatively low risk of adverse effects if the cyanobacteria is less than 20,000 cells per mil. And this would equate to 4 micrograms per litre of microcystin. For moderate probability of adverse effects, it would be 100,000 cells per mil, which would equate to 20 micrograms per litre of microcystin. And a high probability of adverse effects if you can actually see the scum and you can actually visibly see the cyanobacterial bloom in the water. In terms of toxin analysis, there's a variety of methods. And with drinking water, there are provisional regulations of 1 microgram per litre for microcystin LR, which was a World Health Organization recommendation. And with the new Water Framework Directive to be implemented by 2026, this may become then actionable as an action limit. In Australia, they also monitor for anatoxin and anatoxin A at a level of 3 micrograms per litre. And interestingly, when we're looking at some of these algal dietary supplements that you can buy in supermarket shops, they also have a recommendation of 1 part per million of microcystin because some of these algal supplements that aren't really regulated to any great extent by the pharmacy industry or the food industry have been produced from cyanobacteria. Now, the tolerable daily intake as a provisional is around 0.04 micrograms per kilogram per day. And that's based on World Health Organization information. So if you have less than that level per day, it's deemed, that you're at a very safe level. When we talk about methods of analysis, I'm just going to focus on 2 methods of analysis. There's a cost-effective detection or rapid analysis, and we've seen throughout COVID how the lateral flow device changed the face of COVID in terms of being able to determine whether a person was affected at home and whether then they can take instruction to stay at home based on, whether the test was positive or negative, or whether then the test was negative, then it meant they were free to mix with others, go out into the environment, without the fear of spreading COVID. Well, this principle has been taken a slight step further, whereby in collaboration with Queen's University, a company has developed a multiplex test for being able to detect different cyanobacterial toxins that would occur with different algal blooms. And this particular test can detect microcystin and slender spermopsin. The test can take 20 minutes and it can be done at the shore side. So you can actually detect on site at the lake in terms of toxin detection. It's unambiguous in terms of its digital output. We get quantitative results in terms of the level of toxin that would be present. And of course, it's very simple for end users to operate, and perhaps in the future can become part of a citizen science project for Lough Neagh. It also comes with a reader device, which allows the quantification and the simultaneous detection of these different cyanotoxins, particularly the microcystin toxins and slender spermopsin. The result can be in as little as 5 minutes. You just add your sample, walk away, do the reading, and it's low cost and a multiplex test. In terms of the laboratory-based methods then, where we maybe want a full toxin profile for detection, these tend to be much more cumbersome methods. They also tend to be much more expensive methods to run, where the mass spectrometer that actually runs the analysis can be very expensive and crippling for some labs to actually be able to implement. We have to do extraction of the cyanotoxins from the cyanobacteria by disrupting the cells. We have to do centrifugation steps, and then we have to do the actual liquid chromatography mass spectrometry analysis. And the liquid chromatography mass spectrometry analysis, we can use it in 2 modes. We can use it as a screening mode to look for a wide range of toxins at a qualitative level. And we can look at it at quantitative mode, where we look for targeted analysis of particular toxins that we're interested in, and we can do quantification. But we can do a full profile detection of all the different cyanobacterial toxins that could be present, right from anotoxin to homoanotoxin, slender spermopsin, deoxyslender spermopsin, nodularin, guanotoxin, the different range of microcystins, which there can be actually up to 90 different variants, the guanotoxins, and even saxitoxin itself. In terms of strategies of management, well, in terms of mitigation, dealing with an existing bloom and taking the steps to reduce any negative impacts is the term for mitigation. And really, as part of the mitigation strategy, we should really be doing routine monitoring programs, both of the actual bloom and the cell counts, and also of the toxin profiles, and the detection of toxins that leads to the water use restrictions. There's also prevention strategies and actions taken to keep harmful algal blooms from happening or impacting a particular resource. And that would be the control of sewage or waste discharges, the control of nitrogen compounds, chemical fertilizers or fossil fuels, and also the control of land runoff. Now, when we talk about control strategies of management, there's actions taken to destroy HABs to directly intervene in the bloom process, or if not to destroy HABs, to actually prevent HABs from occurring. And there are 5 different mechanisms that might be considered. They might be mechanical, biological, chemical, genetic, or environmental methods. If we think, and we heard of some of these from Mr. Moore in his original opening presentation in relation to some of the SBRI funded projects. If we think about mechanical, it's the removal of the cells from the water by dispersing, in some cases, clay over the water surface, but then there are concerns over the toxin release as the actual cyanobacterial cells start to die. We have biological mechanisms whereby organisms that could control harmful algal blooms. And in terms of these particular organisms, there's some research in one of the SBRIs that I'm involved in for the use of phage or phage bacterium to control cyanobacteria. So the phage would be a virus, and the virus would kill off the cyanobacteria. But of course, there's opposition to the concept of releasing one organism to control another without knowing the full biological effects of that particular organism. In terms of chemicals, even if we add chemicals, again, we have to be mindful that these chemicals will kill the cyanobacteria and release toxins. When we think about genetics, we have genetic engineering of species that are purposely introduced to alter the reproduction or other processes in the undesirable species. But we have to bear in mind that these cyanobacteria are actually the bottom feeders for a lot of our food chain. In the environment, we have the manipulation of nutrient levels in coastal waters, dredging or opening of channels, aeration, and other methods to disrupt stratification. And one of those mechanical methods I would just like to finish with. I came across Dr. David Fung from EM Fluids, and he came to Queen's University. He's a Canadian researcher. He came to Queen's University to give a talk on manipulating thermodynamics of natural systems for cost-effective water remediation and sludge management in large water bodies suffering from eutrophication. And that's the website of his particular company. He's a well-known entrepreneur in Canada, and him and his company have constructed this device which acts as a buoy, and it has an equilibrium modulating stimuli, which creates ripple effects to prevent blooms actually occurring. And the deployment of this device is actually very simple. It has its own battery power, it has its own minimal weight, and it can be deployed in water bodies. And he has shown evidence from different global locations in Thailand, Brazil, and also in other parts of Europe, particularly in Spain, of where this device has worked to prevent algal blooms from actually occurring. And if you want to know more on this technology, I'm happy to discuss with anyone thereafter. So just to summarize, cyanobacteria are a natural part of the ecosystem. The ecosystem is out of kilter to form algal blooms. Climate change has a significant part to play, and algal blooms can produce toxins detrimental to animal and human health. Early detection monitoring programs can be implemented. Prevention strategies are urgently needed that mitigate the blooms, but that do not destroy the wider ecosystem. Mechanical solutions may be the best way forward in the short term, with long-term environmental solutions being sought. And biological solutions can also be a way forward, but need careful introduction to the wider public to be deemed acceptable, even for trial. So I'd just like to thank you for listening, and I'm happy to take any questions that anyone might have.
Thanks, Katrina. We're going to move on to the next speaker just because of time, and we'll have questions at the end, if that's okay with everyone. So reminder, please pop your questions in the Q&A. And if I can now invite Ellie Mackay, who's a freshwater ecologist at the UK Centre for Ecology and Hydrology. Ellie's work covers a whole range of issues associated with water quality, including the causes and consequences of algal blooms in lakes. And she uses a whole range of approaches from traditional monitoring to sensors to understand the physical, chemical and biological changes occurring in lake ecosystems. But she also coordinates and verifies the records for the citizen science app Blooming Algae, which is a tool for recording bloom occurrences and providing information to water users about some of those risks that Katrina's covered today. So citizen science is really something that's close to my heart and close to the work of the Rivers Trust movement. We see communities having a real role here. So very excited to hear your presentation. Over to you.
Thank you. I'll just share my screen. Hopefully you can see that. Yes, thanks, Michelle. So my talk today is really an introduction to the Blooming Algae app. And this is a citizen science initiative, which is really trying to use citizens to provide a surveillance system for where we're seeing algal blooms occurring in lakes and rivers. To transition. So the overview of the talk is really a few questions. So I think Katrina's covered most of this in loads of detail and really interesting, so I won't dwell on them, but really just a reiteration of what we mean by harmful algal blooms and why they matter, why we need to observe them and find out where they're occurring. And then I'll give a brief overview of the Blooming Algae app and how it provides lots of different information on algal blooms. And then a little bit around what some of the data that we're receiving from those records is showing us, and then who else is using that data and what they're taking from it. So so Katrina covered this in a lot of detail. This is what when we're talking about harmful algal blooms in this context, we really mean the cyanobacteria. So and very often what we're thinking about here are the sort of free floating cyanobacteria. So the images on the right-hand side there are from light microscopy, which is the kind of traditional method for identifying and looking at blooms. And what you can see is these organisms grow in a variety of different ways. We get single cells, they can grow as long filaments, and they can form dense colonies. But one of the really key features of those planktonic free-floating cyanobacteria is that they have this ability to regulate their buoyancy. And this means that they float up to the surface and they can form these really dense surface blooms, which we often refer to as scums. And again, the key point here is that the cyanobacteria in particular can, under certain conditions, produce toxins and release toxins into the environment, which can have serious human and animal health effects. And there's been mention today around the issues with dog deaths around Lough Neagh, and that's a regular occurrence. You get records from local newspapers every year of where dogs have been interacting with water where there have been blooms, and unfortunately they've been exposed to those toxins and died. So so that it's a really, really important health risk that we need to be more aware of, and that's where we need to sort of think about it. And in going back to what Katrina was saying around the World Health Organization, this kind of health risk area, where the Blooming Algae app is really useful is that it's really helping us to understand where these scums are forming, because that's kind of the main basis for reporting there. And obviously this is when the category of the highest health risk that are being posed to people. And that information from the World Health Organization is then used by other authorities like the Scottish Government to provide guidance on when and where to monitor water bodies and understand the public health issues around cyanobacterial blooms. So why do we need to record blooms using a citizen science approach? Well, actually, a lot of conventional monitoring methods aren't great at detecting when and where these things are occurring. Typically, we'd be sampling in one location at a lake site or in a river. And these things vary a lot over space and time. So you can get blooms forming under really calm conditions, or blooms being blown into sheltered bay areas. There was some mention of that this morning as well. And if your monitoring point doesn't happen to coincide with where that bloom's forming, then you're not going to see that it's there. So these scums can be really hard to detect because they don't always persist all day either. If you get some wind pickup during the morning and into the afternoon, they can disperse and disappear again and be mixed back into the water column and not be so visible. And so understanding when and where they're happening is really helpful. So some of the sort of key questions that we can ask about algal blooms is, what are the key conditions that are leading to the formation of these scums that we're seeing? Even really simple questions about, how many blooms are there out there? Do we get them every year? How many water bodies are being affected? Are particular water bodies at more risk than others? So those are sort of kind of on the science side of questions that we could ask, but also the really helpful thing about this surveillance system is that we can let other water users know about the risks that happen to them if they're going or accessing a particular water body. So it provides a kind of information and early warning that people need to be aware of these kind of things at a particular site that they might visit. And this information about when and where blooms are occurring is also really helpful for local authorities and landowners, because they can then take action to potentially inform people, put up signage just to make people aware of the potential risks, but also trying to understand what are the causes of those blooms and why they might be happening at that location. So a bit more about the Blooming Algae app in detail. So in addition to being a really great way of recording where we're seeing blooms around the country, it's also been designed to be a bit of an educational tool and provide information to users on what is and isn't an algal bloom. So there's a really helpful photo guide within the app, which provides information on sort of classical features of what to look for in terms of algal blooms, and potentially the things that they can also get confused with. So very often when we're verifying the records being sent into the app, we can get things submitted to us which aren't actually algal blooms at all, and very often they can be things like large accumulations of things like floating pond weeds or just harmless filamentous algae, and they can be indicators of water quality in their own right, but they're not really the focus of what we're looking at here, which is the cyanobacteria that have this toxin producing risk. So when it comes to reporting a bloom on the app, there's a really straightforward way of doing that. You press the record button, and then it brings up sort of a sort of simple table that can be filled in. The key point to verifying is providing us with a really helpful photograph of what you're seeing at the location. The app will use your smartphone's location information to tell us where that is, and you can verify that that's the right site. And then you can tell us a little bit more detail on what's happening at those particular locations. So a kind of quick guide to key advice for that we need when providing a record. Really, to allow us to verify them accurately, we need good images. So ideally, it'd be nice to have an overview of what the bloom looks like, a kind of context kind of picture, to let us understand the kind of scale of the bloom or the scum that's being seen, and then also some close-up images. And that's really key for distinguishing between something that might be a cyanobacteria and something that might be a pondweed or a filamentous algae, and they're quite critical in order for us to verify the records correctly. Then the additional boxes that you're asked about are really helpful to provide a bit more context in terms of how big that bloom is from where you're standing on the shoreline, but also the activities at the location. And that's really important because that helps us to understand the kind of potential risk of different users of water bodies that might be posed by the cyanobacteria being there in the water. So you can see the pie chart on the right there is essentially just a really quick overview of all the confirmed records that we've had submitted to the app, and the different categories that people have submitted in terms of what the water body that they were visiting was being used for. And you can see there from the list of different activities that some of those users are likely to be more exposed to the risks of cyanobacteria than others. So we had talked before about the open water swimmers, and people actually being in the water. It's much more of an issue than people who are just on the shore. Another really useful feature of the app is the ability to view other people's records. So there's a map icon that you can navigate to, and that allows you to zoom into a place of interest. Maybe that's where you go for your walk or your swim, and you can go and see where someone might have recorded blooms previously. It's also useful if you're at a shoreline and you see something and you think, oh, what's this? Has anybody reported this before? You can go on the app and have a look at whether people have previously reported blooms, because what we quite often find in many water bodies is that you'll get a scum forming in one shoreline at one point in time, and then the wind direction changes, and actually that might move around the lake and move to a different location. And so actually having an understanding of some of the dynamics of those changes in the blooms is really helpful to try and understand something more about the potential risks that they pose. So alongside the app, we also have the Blooming Algae website. So I'd encourage you to go and have a look on there. So that provides a bit more information to people, but also what you can do there is instructions on how you can get notifications of when other people are submitting bloom records. So if you sign up with iRecord, you can get a species and notification alert for a particular county or part of a country, or wherever you're interested in. And then every time you get a confirmed verified record of a cyanobacteria being submitted, that will then send you an email alert to let you know that that particular area has had a record confirmed. Other information on there that might be useful to water body managers or owners are some information posters that can be downloaded and printed out and displayed, just to provide more information on the potential risks and things to look out for the general public or people using a particular water body. And then another really helpful feature of the website is the ability to go into an interactive map to go and have a look at where we're seeing blooms. So that's split out by the different countries in the UK. And the example on there is for Scotland, and it allows you to look at different time periods and zoom into particular water bodies and get a really good idea of where and when blooms might be occurring over time. So moving on then to look a little bit more at the records that we've received, and so what we can see is that the app was launched back in 2017, and over the last few years we've seen this really big increase in the number of records that we're submitting, and that's a real increase there in observer effort. So more people are starting to use the app, and we're receiving more records, which is great. And then what we can also tell from that data is that actually as people take up the app and start using it, that actually the proportion of correct records being submitted to us is also increasing over time, which is good. And I think that's an indication that as people get more familiar with what they're looking at shorelines and in locations, that they are more confident in submitting a record of something that's actually a cyanobacteria scum rather than one of these other potential kind of growths of filamentous algae or pondweed. Looking at the same batch of records, so this is just all of the submitted records that we've received over the time period that the app's been operating. You can see a real seasonality in the timing of when we're getting those records submitted to us. So most of the bloom records are occurring over the summer, and that's for a couple of different reasons. So the cyanobacteria are generally more prolific in the summer. They like warmer, stable water columns, and they'll grow really well in there. And they tend to be kind of the dominant. They can be in some sites with the dominant algae within the phytoplankton community, that open water algal community at that time of year. But also the summer is also when we like to go and spend time by watersides and use water bodies, and so it's probably also an indication of kind of when people are most likely to encounter these blooms, and the two kind of coincide there. But what you'll also notice is actually that we're seeing blooms occurring throughout the year, and we get records. I was verifying some this morning, actually, from a couple of sites in England. So there are reports of blooms coming in all year round, but just generally a lot less common outside of that kind of sort of late spring, summer period. And going back to kind of what Katrina was saying on the climate change potential impacts on blooms, where we've got a lot of nutrients within water bodies. The potential increase in temperatures that we're likely to see, and the effects on rainfall patterns and other things, it likely to mean that we're getting more records during the summertime, but also potentially a spread of records occurring more at other times of year as well. So a real potential for an increased risk of blooms occurring. So whereabouts is the app being used? So over the sort of since 2017, the app was initially launched for the UK, and then since then, it's other sites have been added. So we now receive records from Ireland, the Netherlands, Norway, Belgium, and Luxembourg. So you can see there that kind of Northwest European map where all our records are coming from at the moment. But also we're sort of expanding, and thanks to some funding from an EU project, we're adding new countries in Denmark, Finland, Greece and Poland. And then through some international funding that we have within UKCEH, we're adding in Chile and Kenya, which will be great. So we've got this expanding coverage across the world, and what you can see is that clearly, where as soon as we start taking records from other places, that this is not a UK problem. This is definitely a global issue, and it's useful to try and understand more about when and where these blooms are occurring. So who's using the data? Well, there's sort of various different groups who make use of the data. So the app users themselves find it really useful to understand when and where blooms might be being reported on water bodies that they use themselves. Landowners use the information to help them put up signage or understand if there are potential risks to water bodies where there might be activities occurring. We also work really closely with some environmental regulators. So SEPA in Scotland have been really big supporters of the app, really from its inception, and now they have members of their teams go out and record blooms that they're seeing out in there, and SEPA themselves act as verifiers on the app, so they're aware of when people are recording blooms through blooming algae in Scotland. And similarly, for the last couple of years, we've also had colleagues in Dira in Northern Ireland, both reporting blooms that they're seeing when out and about, but also acting as the verifiers for those bloom records. And that's great, because that means there's a real connection between the kind of bloom records being observed, and then the people in the regulatory agency has been aware of what things are happening and when. And similarly, the Environment Agency in my local area in Cumbria utilise the notifications that come through the app to add that information to their blue-green algae pages that they host each summer. Back in Scotland, Public Health Scotland have been actively using the data on records that we've been receiving from SEPA, but also produced a new public health guidance on cyanobacteria back in the summer last year, which advocates for the use of the app to help verify that blooms are occurring and as a way of reporting them. And increasingly, we're seeing more researchers take up the bloom and algae data to start trying to understand more of those kind of fundamental science questions around the occurrence of these blooms and understanding where they're happening. So to sum up then, really, blooming algae as a citizen science tool is really helpful to inform risk management of algal blooms. So it can provide support to agencies and authorities and provide rapid warnings to reduce those public and animal health risks that are being posed by blooms occurring. They can potentially be used to reduce monitoring costs. So some of those regulators, now they're verifying the blooms themselves. They don't have to go out and take a physical water sample to analyse it and confirm the sightings. If there's a scum present in the water body, then that's clearly a high risk of there being toxin potentially there, although that's not the same as measuring the toxins itself. So the app can also help to trigger operational decisions. So it might help to inform where you might want to go and do some additional monitoring to look at toxin concentrations, for example, or risk management actions. So trying to mitigate risks of potential water users, or look at trying to understand, what the causes of the bloom that might be occurring at a particular site is. And then finally, it's helping us to provide us with more scientific evidence on the drivers of these blooms, and linking that data up from the app with other data sources that can help us to try and understand better when and where blooms are occurring, and what we can try and do to mitigate that most effectively. So how can you get involved? Well, I'd encourage you to download the app and record any blooms that you're seeing. You could sign up for bloom alerts in your area if you're a regular user of a particular water body, and you can use the information posters from the website to inform other people of the risks of cyanobacteria blooms. So with that, I'd just like to say thank you. This is obviously a big team effort. So there's a team of us in UKCEH involved in the Blooming Algae app, but also our colleagues in SEPA and DERA, and also Lawrence Carvalho at NEVA now, but he was a real driving force in getting the app set up when he was at UKCEH. So with that, I'll say thanks, and yeah, happy to answer any questions.
Many thanks indeed, Ellie. I think that's a really encouraging overview of something that everyone can do to help play a part in this. If I can please invite Katrina as well to turn her camera back on now. And thank you, Katrina. You've been busy answering some of the questions that came in the chat, including, there was a really good point from Ellie Knowles about what role can we all play in not transferring this problem from one water body to another. And I think, I don't know, Katrina, if you want to talk briefly on that one, because I think it's really important for people to be aware of this.
I think it's, from one water body to another, it's really important that we...
You're a bit quiet, Katrina, if you can lean in. Sorry, I don't know if that's just me or...
Okay. I think it's very important in moving from one water body to another to make sure that the gear is well cleaned. Whatever gear has been in the water, whether it be clothing, whether it be equipment, that it's well cleaned. And as Knowles actually came back with was the clean, check, dry approach, maybe CCD approach, if you want to shorten it. But the clean, check, dry approach before you move to other water bodies, so you don't have cross-contamination going from one lake or loch to another, or one river body to another, would be very important.
Yeah, just as important for invasive species as it is for blue-green algae. Thank you.
Sure.
We've got a couple more questions in the chat. I'm going to pick one. We've got one from Bethan Edmonds. Is it possible to tell whether the bloom or scum layer is caused by a toxin-producing bacteria, or does it have to be tested with one of the methods that Katrina mentioned? Is it safer to assume that it's toxin producing in the first instance? I don't know, Ellie, if you want to come in with that one.
Yeah, I think essentially the regulatory agencies take the approach of being risk averse. So if you see a scum, then it's a high risk of potentially holding a toxin, but that isn't the same as it actually being a toxin producing strain. And I think you'd have to take a sample and analyze it. And obviously Katrina mentioned the kind of bankside analyzers, which are starting to come online, but I think the issue with toxin production is that actually the mechanisms behind all of that, there's some understanding of that, but I know that there are some other people on the call who I've heard talk about this before, that you can measure a sample 1 minute and it has no toxin within it, and then several hours later or whatever you might find that there's toxins present in the water. So I think generally people take a risk averse approach and don't go into water where there's really obvious bright green scums.
I think there's a role for both, the sort of science that you were talking about, Katrina, in the labs, but also people, because it's so temporally variable, getting eyes and ears on the ground, which is where citizen science comes in. And Ellie, there was a question on, is there any plans for training on the system, the eye record systems of human algae? Will there be any sort of training programs that you're aware of, or is that something we could look at in the future?
In terms of using the app, or in terms of verifying the app?
I think, people, it's from Claire Crossan, and they're using iRecord to verify submissions of blue-green algae. So I think they're looking for training as the verifier.
Oh, okay. Yes. Yeah. So, we're hoping to organise some sessions for potential verifiers later in the year. So if you get in touch, we can get you on the list, and we can then let you know when that's going to happen.
Thank you. Okay, well, with that, I'm afraid we're out of time, but there are plenty of questions still in the chat if the speakers have any more time to follow up. But I'd just like to thank you both for your really enlightening presentations today, and hopefully we've all learned something on their own. I'll hand back to Becca. Thank you.
Thanks, Michelle. Thanks, Ellie, and thanks, Katrina. Really, really brilliant content there. I was going to use the opportunity as a lovely segue to give another shout out to our Big River Watch app and campaign. I see that Joanna has partly done that job for me in the chat, but even so, I would encourage everyone to follow that link, download the Big River Watch app and get involved by recording observations of your local river. That is between the 25th of April and the 1st of May. It's really accessible and it's just really important for us to better understand how you interact with your local river. So please send in any questions on that about how you can get involved. Otherwise, I will let everyone take a well-deserved lunch break, and we'll be back in an hour for a more solutions-focused session with Rob Collins and some other speakers. So thanks for sticking with us so far, and we'll see you at 2 p.m.